Let us now move on to nanomaterials. So nanomaterials, they are defined like this: materials with particle size in the range 1 to 100 nanometers. Normally, for materials, the particle size will be in the range 1 to 100 micrometers. Okay, but if you can reduce the particle size to 1 to 100 nanometers, then that is called as a nanomaterial. Nanomaterials, they have some unique properties compared to normal materials. We will be seeing that a little later. How are nanomaterials classified? That is what we are going to see, the classification of nanomaterials. So first classification is based on the dimension. So based on dimension, we can classify a nanomaterial into 0-dimensional, 1-dimensional, 2-dimensional, and 3-dimensional. 0-dimensional nanomaterial, 1-dimensional nanomaterial, 2-dimensional, and 3-dimensional nanomaterial. What is a 0-dimensional nanomaterial? If all the dimensions, that is for a particle, there will be 3 dimensions in the x, y, and z axis. So if all these dimensions are in the nano size, nano range, that is 1 to 100 nanometers, then that is called a 0-dimensional nanomaterial. For example, fullerene. 1-dimensional means one of the dimension has measurable length, that is, it is in micrometers. For example, carbon nanotube. Here it's a cylindrical tube, so the diameter is nano size, but the length is in the micrometer range. So it is a 1-dimensional nanomaterial. 2-dimensional means 2 dimensions have measurable length. For example, graphene sheet. Graphene means it is one layer of a graphite sheet. Graphite has a layered structure, sheet structure. If you take only one layer out of that, then that is called graphene. Graphene is a 2-dimensional nanomaterial. And if all dimensions are measurable, then that is called 3-dimensional nanomaterial. For example, box-shaped graphene. So this is how nanomaterials can be classified based on dimension. There is one more classification which is based on content. Okay, what is the content in a nanomaterial? So based on content, we can classify nanomaterial into carbon based, that is the first category. Here the content is carbon. Then metal based. Here the content is metal or metal oxide. Like for example, gold, silver, copper, etc. Metal, they can be nanoparticles, or oxides, aluminum oxide, titanium oxide, etc. Nanopolymers. So polymers, normally they are not nanomaterials, but there is a class of polymers which is called a dendrimer. These are nanoscale. These are in the nanoscale. Nanocomposites. Composite means a combination of more than one materials. So if in a composite, if one of the material is of nano size, then that is called a nanocomposite. For example, polymer filled with carbon nanotube. And finally, biological nanomaterials. Nanomaterials of biological origin. Example, peptides, DNA, then enzymes, etc. etc. So these are the classification based on content. So one important question, discuss the classification of nanomaterials. What are the unique properties of nanomaterials? So most important property, it has unusual mechanical properties. Mechanical property means something like tensile strength. That is a mechanical property. So unusual tensile strength. Okay. Then high electrical conductivity. Example, carbon nanotubes. They are found to have very high electrical conductivity. Large surface area. When the particle size decreases, surface area automatically increases. So large surface area. More reactivity. Nanomaterials are very reactive. They can combine together to give a micromaterial. Okay. And high thermal stability. They have very high thermal stability. So these are some of the unique properties of nanomaterials. Again, enhanced catalytic properties. Since they have large surface area, they can be used as good catalysts. Synthesis. How can we synthesize a nanomaterial? There are many methods for synthesis of nanomaterials. We will be dealing with two important chemical methods. Okay, two important chemical methods, that is reduction and hydrolysis. Reduction and hydrolysis. Let us see one by one. Reduction, you are reducing a substance into a nanomaterial. Reduction can be of two types. One is chemical reduction, the other is electro reduction. Chemical reduction means reduction by adding chemicals, and electro reduction means reduction by passing electricity. So we can see that one by one. Now chemical reduction, for example, if you want gold nanoparticle, then you start with gold chloride solution, and to this gold chloride solution you add a reducing agent, that is sodium boron hydride is the reducing agent. So you add the reducing agent with vigorous stirring, and then you add a stabilizer. Stabilizer is citrate ions. So that will give you the gold nanoparticle. Citrate ions are added so that the nanoparticles will not combine together to give a larger particle. Citrate ion will act as a spacer between them. Okay. So this is how we can synthesize gold nanoparticle. For example, this is gold plating solution. Gold solution is yellow in color, but when it is converted to a nano gold, it becomes red in color. The properties are entirely different. Coming on to electro reduction, here again you have a, let us take the case of silver nitrate solution. You take and you insert electrodes into that and pass a current. So silver nanoparticles will deposit on that electrode. You can control that. You can control, if you control the current applied and you change the concentration, we can control the nanoparticle deposition. So that is electro reduction. Next is hydrolysis. Hydrolysis, again there are two types of hydrolysis. One is called sol gel method and the other is called hydrothermal method. Sol gel and hydrothermal, both are similar. Both are similar. Now what is happening is you are first preparing a colloidal solution of a substance called a sol. This is a sol. It's a colloidal solution. Then that sol is converted to the gel. It is converted to the gel. What is a gel? A gel is a thickened sol. Okay, when the colloidal solution thickens, it does not precipitate down, it just thickens. That is called as a gel. Okay, so you are preparing a sol and you are converting into a gel. Depending on how you do that, we get these two methods, sol gel and hydrothermal. Essentially they are similar. Let us look at each in detail. So sol gel method, let us take the case of aluminum oxide. You want to prepare aluminum oxide, so you start with aluminum nitrate solution. To this aluminum nitrate you are adding a precipitating agent, that is ammonium hydroxide, with vigorous stirring. So add ammonium hydroxide slowly with vigorous stirring, you get the aluminum salt, aluminum hydroxide salt. Then this salt, it is kept without shaking, it is kept for a few days. That is called aging. It is aged for a few days, you get the gel, aluminum hydroxide gel you get. and then this gel is dried and it is calcined. Calcined means heating at high temperatures, above 300 degrees. If you are heating, that is called calcination. So drying is at 100 to 150 degrees centigrade. Calcination means above 300. So when you heat, it will dry and it will get converted to the aluminum oxide nanoparticle. So this is how we can synthesize aluminum oxide nanoparticle by salt gel method. Okay. The next one is hydrothermal method. How this is done? The steps are similar. There is only a small difference. The salt converting to the gel, that part is little different. Okay, let us take a case of silicon oxide. Suppose you want silicon oxide nanoparticle. Then the starting compound is this one, tetraethyl orthosilicate. Okay, tetraethyl orthosilicate. So that is dissolved in isopropanol. And to this we are adding a precipitating agent. Here the precipitating agent is water. Add water slowly with vigorous stirring, you get the silicon hydroxide salt. Silicon hydroxide salt. Then what you do? Transfer this salt into a container, a sealed container, and heat it. Okay, transfer this salt into a sealed container, stainless steel container, and heat it. So when you heat that, it gets converted to the gel. This is called hydrothermal. Heating in a sealed container. Hydro because we are doing it in aqueous medium. So it is called hydrothermal. Hydrothermal synthesis means heating in a sealed container. Okay, so you get the gel. The gel is then dried and calcined to give the silicon oxide nanoparticle. So this is how we can do hydrothermal method. So one important question, describe any two methods for the preparation of nanomaterials. Okay, reduction and hydrolysis, both are very important. What are the applications of nanomaterials? Many applications are there in batteries as electrodes, then catalysts and supports, then lubricants, electronic and microelectronic devices like chips or capacitors, etc. Then drug delivery and tissue engineering. These are some of the important applications of nanomaterials. Now, how do we find out whether a substance is a nanomaterial or not? That is the important thing. You are given a substance, you don't know whether it is a nanomaterial or not. But look, just by looking at it, we cannot find it out. So we need to have some techniques, some instruments to find out whether a substance is a nanomaterial or not. And one such instrument, that is called the scanning electron microscopy. Okay, scanning electron microscopy. So microscopy means what? Microscopy means you are magnifying something, okay, using lenses. Using glass lenses, we are magnifying something and we are viewing it. Okay, and in this we are using visible radiation. You can see it. So we are using visible radiation. So in scanning electron microscopy, what you do is, instead of visible radiation, you are going to use an electron beam. Electron beam. Okay. So the principle is like this: producing an image of a sample by scanning a surface with a focused beam of electrons. You have an electron beam. So electron beam, when you are using, you cannot use glass lenses, you have to use magnetic lenses for that. Okay, so using magnetic lenses, using electron beam, we are getting an image, we are getting a photograph of a substance. A magnified photograph of a substance is obtained. Okay, what is the advantage of that? we can get information about surface topography and composition. Surface topography means what is the particle size, what is the distribution of the elements. All these things we can understand. Everything about the surface. A geography. Okay, how is the surface? Is it a plain surface, or is it porous in nature? Are there holes on the surface? Everything we can understand. Now let us look at how this instrument works. The instrumentation. So this is a block diagram of the instrument. Now you can see here, this is an electron source. It is called electron gun. It produces high energy electrons. And using this anode and cathode, what you are doing is you are allowing these electrons to move in this direction. Okay, it is allowed to move in this direction, and there are some lenses. You can see an objective lens, you can see condenser lens. All these are magnetic lenses, and using these magnetic lenses we are converting this beam into a narrow beam. Okay, narrow beam, and that is allowed to fall on this plate on which you have coated the sample. The sample is coated on a square plate. Okay, now here there is something called XY scan coils. What does this XY scan coil do? It allows this beam to move in the XY direction. So this is a square plate of something like 1 centimeter by 1 centimeter area, 1 centimeter square area. So this XY coil will make this beam move in that 1 centimeter square area. Okay, it will scan that area. It is not coming as what you call standing at a particular point. It is moving. It is scanning this entire area. Okay, then what will happen? You are sending in high energy electrons. These electrons will interact with this sample and it will produce something. Based on that, we can get information about the substance. Okay, now what does it produce? These are the three things: secondary electrons, backscattered electrons, and X-rays. These three things are produced. Okay, let us look into that detail. So let us look at the working. Electron gun emits a beam of electrons. It is focused using a combination of condenser and objective magnetic lenses. The focused beam passes through pairs of scan coils which deflect the beam in the XY plane. The electron beam falls on the sample and interacts with the atoms on the surface. Three types of interactions are possible. Now what are these three types of interactions? The first interaction is inelastic scattering. Inelastic scattering means scattering by change in kinetic energy, and that produces secondary electrons. Okay. Second one is emission of X-rays from the atoms. These atoms on the surface, they can absorb these electrons and they can emit some X-rays. And third one is elastic scattering. These electrons, they come, they are reflected without any change in the energy. That produces backscattered electrons. Okay. Now what information can we get from these? Secondary electrons. If you analyze the secondary electrons, we get information on the topography, surface topography, particle size, etc., etc. If you analyze the X-rays, we can get elemental composition, because each element will release or will emit one particular X-ray. For example, silicon, it emits one particular X-ray. Oxygen emits another X-ray. So each element will be emitting a different X-ray. So we can understand about the composition. And finally, backscattered electrons, they give the distribution of the elements on the surface, how these elements are distributed. So this is how this technique is working. So this is how we can get information about a particular substance. Okay, applications. What are the applications of this technique? We can detect and analyze surface morphology of almost all compounds. Then we can examine surface contamination, whether the surface is contaminated, whether a substance is contaminated. For example, when we are preparing what we call semiconductors, if any contamination comes, then the working of that semiconductor will change. Okay, so we have to be very careful during the synthesis, so we can check whether a contamination has taken place. Determining chemical composition, we can determine what is the chemical composition of a substance, of unknown substances. We can find out, then we can identify crystalline structures, for example like cubic structure, orthogonal, hexagonal, etc., that we can find out. And this is widely used in life sciences, medical sciences, forensic sciences, and metallurgy. So these are some of the applications of scanning electron microscopy. Let me show you some images. Okay, this important question: briefly explain the principal instrumentation and applications of SEM. This is asked as an 8-mark question. Now this is a structure, rock salt, sodium chloride structure. Okay, it is a cubic structure. This is pollen grains, the SEM of pollen grains, and this is the SEM of sucrose, sugar. It has hexagonal structure. Okay, hexagonal structure. This is human hair, and this is human hair with dandruff. Okay, so these are some examples of scanning electron microscopy. So this is our topic, nanomaterials and scanning electron microscopy.